Growth and development in plants are regulated by some special chemical substances are called phytohormones or plant hormones. These phytohormones, as termed by Thiemann in 1948, plant hormone is an organic compound. It is synthesized in plant, one part of the plant in minute quantities. From there, it is translocated to another part where it causes the physiological response. Chemically, they may be indole compound IAA and IBA. Adenine derivative is cytokinin. Derivatives of the carotenoids ABA. Terpenes gibberellic acid. Gases ethylene. These are easy to synthesize, easy to transport. They may be show effect in minute quantities and presently known to regulate the growth. They can be placed into two groups based on their function. Growth promoters, auxin, gibberellin, cytokinins, brasinosteroids. These are promoters. Second one, growth inhibitors. Ethylene, abscisic acid, jasmonic acid is these all are growth inhibitors. Auxins. It was the first growth regulator to be discovered. These are indole derivatives. Chemically, these are organic compounds synthesized in the stem apex and root apices. From here, they migrate to their sites of their action. Auxins are synthesized from the tryptophan. Let us talk about the discovery of the auxins. So, in 1800, Darwin and his son Francis did experiments about when they were studying the phototropism in plants. He did experiment with the Phalaris canariensis, in general canary grass and oats. Charles Darwin and his son both together worked on that. They carefully observed that when the coleoptile exposed to the unilateral light, the bending towards the light can be seen. Whenever the coleoptile cut, there is no response towards the light. Eventually, there is a conclusion between that. Some substances synthesized in the tip of the plant and it is transmissible to the lower parts of the plant. In 1907, Fitting did the experiment with the saturated water vaporized room and gave incisions on the plant. The plant gave bending towards the light, unilateral light. In 1910, 11, 13, Boyson and Jensen did the experiment with cut coleoptile, then replaced it on the stump. On unilateral light, the coleoptile tip gave the positive curvature indicating that conditions will not interference with the causing the curvature substance. In second experiment, he removed the colloidal tip and placed the gelatin black. Then over that colloidal tip placed on it. So bending is seen even cut the colloidal tip. It clearly indicates that it was the water soluble substance. In second experiment, he cut the colloidal tip and replaced it by the a gelatin black. Then over it again he replaced the colloidal tip. So bending can be seen. It clearly indicates that this chemical substance is water soluble substance. In third experiment on unilateral light side, he inserted a mica plate even though bending is seen. In fourth experiment, he inserted the opposite of the unilateral direction of the dark side. Here there is no bending is observed. Concluded that the curvature causing stimulus is active in dark side of the colloidal tip. 1914 and 1919, Paul did the experiment with the colloidal and cut that tip. Again he replaced on the stump asymmetrically. Positive curvature was observed even it transferred to the dark. In second experiment, on unilateral illumination, he inserted the mica plate and replaced the colloidal tip. There was no phototropic response. It concluded that it is not electrical and not fat soluble. So in 1925 did the experiment. He removed the colloidal tip and afterwards he replaced on the stump. So he concluded that the growth rate is much reduced. On contrary, replacing of this colloidal on the stump again, the growth rate somewhat reduced on comparison to the control experiment. 1928, Went did the experiment with the Avena colloidal. He cut the colloidal and replaced on the agar-agar block for a measured length of time. And again he replaced this agar block asymmetrically on the cut stump. There was a bending seen. It placed on the colloidal stump centrally. The growth was renewed. He called for this substance as auxin. He also found that the curvature of the Avena colloidal is proportional within limits to the amount of auxin in the agar-agar block. Therefore, this test was named as Avena curvature test.